for being here today, and to Carol for asking me to speak here. So I'm going to talk first of all about some data that we've got from an animal model, but the reason I'm showing you that is so that I can try and explain what it might mean, because obviously it's much easier to do experiments in an animal model than it is in humans. So almost everybody, I suppose, that's listening realizes that almost 2/3 of people with Alzheimer's disease are women, and that means that right now there are about 3.8 million people with Alzheimer's disease. We don't know the reasons why this is the case. Obviously, one of the things that has been suggested is that the menopause leads to a reduction in female hormones, estrogen in particular, which we know is neuroprotective. That's one possibility. Other possibilities include the likelihood, or the fact that women are more likely to suffer from depression, which could be a risk factor, more likely to have a particular type of obesity that may be a risk factor, and so on. But the answer is we have no idea why this is. So one of the things that I'm pointing out here is that we have non-modifiable genes, and I know that Stefania is going to be talking much more about this than me. I just want to make one point here. These non-modifiable issues are age, genes, and sex, but there are plenty of modifiable factors that introduce risk of Alzheimer's disease. And the ones that I've highlighted here in red, obesity, diabetes, and head trauma, as well as cardiac health, are 4, plus others that I've indicated here in green. Now, the thing that they all have in common is that they're associated with inflammation in the brain. And the reason why I'm bringing that up is because we now believe that inflammation is one of the earliest changes that takes place in the progression of the disease from normal through MCI and into dementia. And it precedes changes, for example, in amyloid accumulation in the brain, as well as these others: brain structure, memory, and clinical function. So the work that I've been doing for the last several years is to try and figure out, why is there inflammation in the brain in age, first of all, and why is it far more evident in individuals with Alzheimer's disease? And when we talk about inflammation in the brain, we have to consider the cell type that's primarily responsible for it, and that is, I'm having difficulty shifting. They are the microglial cells. So the job of microglia under normal circumstances is to protect the neurons. So what they do is they survey the environment that they're in, and they look out for any damage. When they sense that damage has occurred in any particular area of the brain, they change their shape from this star-like shape with lots of processes to a much more amoeboid shape. This is a roundy shape with far fewer processes, and those processes that are left are thicker and, as you can see, much less extensive. So when the cells are in this shape, they move towards the area of the damage, and the idea is that they will eliminate the problem. So I'm just going to show you a very short video which illustrates this. So these green cells here are microglia, and the processes are these little dots. This is real-time imaging, and as you can see, the dots are moving all the time. Now what's going to happen is that you will see a yellow blotch appearing on the screen, and what that is a stab wound to this blood vessel. You can just about see red there, which is indicating a blood vessel. So the experiment was, this blood vessel was stabbed, and let's just have a look at what happens when this occurs to the microglia. So here's the stab, and as you can see, microglia start clustering around the area where that has occurred. So there's blood seeping out of the damaged blood vessel, and what you will see now is that some of these cells are turning yellow, and that's because they're engulfing cell debris and blood cells that have occurred as a result of the damage. Now what you'll see in a moment is that the blood flow is restored because the cells are protecting and repairing the damage. And so in a minute now you'll see the blood vessel being restored and blood flow resuming through this area. So what I've tried to illustrate with this video is the cells move they change their shape, lose their processes by and large, and as you can see, they start engulfing the damage. And the idea is that they prevent further damage from occurring. Now, the cells are constantly on the go, these microglia, are constantly on the go, hoovering up damaged cells or bits of debris, and also hoovering up amyloid, which is the peptide that coalesces to form the plaques in Alzheimer's disease. The problem is that when we look at these microglia in the brains of this model of Alzheimer's that we're using to investigate them, what we see is the cells become exhausted and they're no longer able to hoover up the damage or the amyloid, and so we get an accumulation of amyloid. Now, the reason why I pointed out that the cells were constantly active was because I wanted to tell you that they need energy all the time. And without being any way detailed about it, energy comes from glucose, and it's broken down. This glucose is broken down ultimately to water and carbon dioxide. And when that happens, you get 32 molecules of ATP. ATP is the energy source. However, sometimes the cells stop at a point where pyruvate is made, and that's glycolysis. And when the stop takes place at that point, we only get 2 ATP molecules. And what we have found is that in microglia from our female mice that are the model mice that we use for APP, we see glycolysis has increased. So the cells look like they're relying more on glycolysis than they are on full metabolism. And as a result of that, less ATP is produced and the cells get tired, in fact, exhausted, and they're no longer able to function the way they should. So in contrast to the female cells, which are these brown ones up here, the male cells are whether they're from wild type mice or from our model of Alzheimer's mouse, they're all the same. So the female cells are much worse off. They're more tired. Now, we started to look at the shape of these cells, and I'm telling you about this because I'm going to try and use the information that I'm going to tell you about now to give you some idea of what's happening in the brains of human individuals that have had Alzheimer's disease. So first thing that we did was we took sections from male and female APP mice. These are our model of Alzheimer's disease. And what we found was that there was a very big difference in the shape. And as you can see here, I've highlighted the fact that in the male brain, we have lots of these cells that have very few processes. These we call amoeboid cells, compared with the female brain, where we have lots of cells that have these processes. And you can see those quite clearly here. What we then did was we looked to see whether or not there was a difference, I beg your pardon, a difference between the ability of these two cell types to function, and in particular to function by means of taking up amyloid. And so here we have two of these amoeboid cells, the kind that you see in males compared with females. And what you'll see is that there's a lot of green and yellow staining inside those cells. And that tells us the cells are taking up the amyloid, which in this instance is stained green. And here's a cell with lots of processes, as you can see. And what we see is there's very little amyloid being taken up into these cells. And in the end, when we analysed all the data, what we found was that in female APP-PS1 mice, the microglia are much less able to take up amyloid than the cells from males, where there's very little difference between the amyloid, the model of amyloidosis and the controls. Now, the other thing that we found when we were examining these cells was, here you can see the difference between males and females. It's very evident. You have these small amoeboid cells in the male, and that's not the case in the females. But what we did find in the females were lots of these elongated cells. These are called rod-shaped microglia. And the story in the literature is that these cells are persistently active, and they're so persistently active that they're troublesome, because these cells actually are responsible for inflammatory changes. And we know about these cells also that they are unable to phagocytose. And this diagram here shows what happens when we counted the cells. And the most important message is to show you that the rod-shaped microglia in the female APP-PS1 mice are much more prevalent than in the males. The other thing we found out about these cells from staining the tissue from females compared with males is that in female brains, you have far more iron accumulation. And the iron is shown here in these darkly stained cells. As you can see, they surround these pink plaques. These are amyloid plaques, and we find, oh, I'm sorry about this. We find that the iron labeled cells, or the cells that have accumulated more iron, are far more likely to surround a plaque than they are to be elsewhere. The other thing that I just want to point out here is that, as you can see in the brains from the male mice that we use as our model of Alzheimer's, there's far less iron accumulation, and they don't seem to be as fond of surrounding the amyloid plaques as the females. And here, when we counted the cells, what we find is, once again, the significant change is in female APP-PS1 mice as opposed to males. Now, what's why do we care about iron accumulation? Well, it turns out there are a few things that are very worrying about excessive iron accumulation. And one is that they drive the cells to become more glycolytic. And here we have the same sort of graph as I showed you before. Glycolysis is increased in cells that have more iron engulfed. And this means that they are less efficient in terms of producing ATP. And in addition to that, these iron-labeled cells, or iron-laden cells, I beg your pardon, increase the production of inflammatory cytokines. And here's one, TNF-alpha. But it's not just TNF-alpha. Other cytokines that we know to be inflammatory are also increased in cells that accumulate iron. And these cells also cause microglial activation. So now I'm going to use that information that I've told you about, and I'm going to now move on to looking at the results that we got when we started to investigate brain tissue from post-mortem Alzheimer patients. So just to recap then, what we find is in the brains of these models of Alzheimer's disease, there's inflammation, the cells become glycolytic, and because of that, the cell's energy is compromised and the cell function deteriorates. In addition, cells accumulate iron, and that could well be responsible for some of these changes. And in the end, when these cells are not able to phagocytose the amyloid accumulates. I made reference to the fact that microglia change their shape, and in males, we have these amoeboid cells that are able to engulf amyloid, and in the females, we have rod-shaped microglia that are more inflammatory. So we got tissue from the Netherlands Brain Bank from individuals with Alzheimer's disease and age-matched controls, and we had males and females that were fairly well matched in terms of their BRAC stage for the Alzheimer patient and aged matched, as I said, for the controls. So the first thing that I want to point out here is that there's a difference in the shape of the cells in individuals with Alzheimer's disease compared with the controls. And here you see very little difference between the control males and the control females. They look pretty much the same. But when you look at the sections from individuals with Alzheimer's disease, there's quite a marked difference. And in particular, what you see in the AD males is that the cells adopt this amoeboid shape that I mentioned to you already in the mice, and they in the females, we have these processed cells. Now this, if we can extrapolate from the mouse, tells us that the cells from the males continue to phagocytose and are more functional generally, but in females, that function is lost, and these cells instead become inflammatory. So the second thing that I want to point out is that when we did our analysis of these cells, in this instance, what we did was measured the circularity of the cells, what we found was the circularity was increased in the sections from the Alzheimer male patients, but not in the female. And remember, I've already said that amoeboid cells tend to be phagocytic. In contrast, in the brain tissue from females, what we find is we get these elongated rod-shaped microglia, and you can see there's a very marked difference between the Alzheimer females compared to the Alzheimer males. And this, we think, might contribute to the inflammation that's already been described in the brains of Alzheimer patients. Now, with regard to the cells, another thing that we found when we were examining these cells from the males versus the females was that in the females, there were an awful lot of cells that looked like they were what we call dystrophic. The cells look like they're breaking up. And here you can see evidence that the cells are beaded, as it's called, and in particular that the processes from these cells are beaded. And when the cells are like this and the processes have broken up, it means the cells are not able to move in the same way as they were able to move under normal circumstances, and therefore they lose their protective effect. And so this is something that's much more evident in females compared with males. And here you can see there are twice as many, if not more, dystrophic microglia in the brains of females compared with males. Even the females that are not in the Alzheimer group, but the controls, but in the Alzheimer group, these are more common again. And the other thing that we found in female brains compared with males is that there's a marked increase in the number of cells that engulf iron. So we have marked iron accumulation, as you can see in cells from females compared with males, and that's shown in terms of numbers on the right-hand side of this diagram. So remember, I suggested to you that from the experiments that we did in mice, when we get iron accumulation, a few things happen. First of all, these iron cells that accumulate iron produce inflammatory cytokines. Secondly, they change their metabolism so that they become less efficient. And thirdly, they become less functional. And so we end up with cells altogether that cause trouble in females to a much greater extent than in males. And the next change that we found in the females that was much more marked than in males is this lipofuscin staining. Now, lipofuscin identifies cells that have accumulated a collection which is made up of lipoproteins, proteins, and lipids. And what it does is indicate to us that there's a problem with mitochondria. And again, this points to the change that has taken place in the metabolism in AD patients. And it's much more evident in females compared with males. There's also a very big difference in the way the accumulation takes place in males and females. In females, it's scattered all over the brain, and even in areas that are not highlighted here, you can see quite a bit of grey, if not black staining right throughout, whereas in the males, wherever lipofuscin staining is found, it's found in a cluster as it is here. The final thing that I want to mention to you about what happens in females compared with males is that the plaque area, this is the area of the brain that's covered in amyloid plaques, is greater in females compared with males. Now we measured, we counted plaques as well. We didn't find a difference in the number of plaques, but what we found was this plaque area that was markedly different in females compared with males. So this all gathers together the information that we have that suggests that one of the reasons why we might have this amyloid accumulation is because the microglial cells, which normally take up the amyloid, are no longer capable of doing that, particularly in female AD patients. And so in conclusion then, what I have to point out is that inflammation is associated with a number of changes in the microglial cells, and they include a change in metabolism. So the cells are now a little bit compromised, and as a result, the function decreases. So the sex-related differences in microglia that we've identified are, first of all, metabolism is compromised, as is function. We get an increase in inflammation, and we see an increase or an alteration in morphology and in iron retention. And finally, I would like to suggest to you that the microglia from female AD patients are dystrophic. This means that they're no longer able to function in the way we'd like them to. They are rod-shaped. That means they contribute to the production of inflammatory molecules, and they're iron retentive. This means that amongst the other things that we might be able to suggest about these cells is that we have mitochondrial dysfunction, and that means that overall the function of the cells are seriously compromised. And so I'd like to acknowledge the people in the lab whose work I referred to, and they're all here in black. And I'd like to point out in particular that Owen O'Neill and Sybil Bechet have been hugely important, as has Virginia, in driving this story. And we had funding from a number of different sources, and very grateful to those funders, as I am to the collaborators. Thank you very much.
Thanks very much, Marina. That was absolutely fascinating and really clearly explained for something that's so complex. I think that came across very clearly to me anyway, and I hope others found that very interesting. So just a reminder, if people have any questions for Marina, you can put it in the chat or the Q&A buttons there on your screen, and we'll have time at the end to look at those questions. Okay, so our next speaker is Dr. Stefania Ilinica. Stefania is a Senior Atlantic Fellow for Equity in Brain Health at the Global Brain Health Institute in Trinity College Dublin. She's also a Senior Researcher at the European Centre for Social Welfare Policy and Research in Vienna, and an advisor for long-term care with WHO Europe. And Stefania is going to talk about gender and socio-economic differences in modifiable risk factors for dementia throughout the life course. Over to you, Stefania.
Thank you very much, Carol, and thank you for inviting me. And of course, yeah, fascinating talk by Professor Lynch. I think it's going to be a really significant change of pace now. And it's fascinating to me as a social scientist to get a glimpse of the complexity of biological systems. I guess I want to draw attention in my presentation on a very different type of complexity that we deal with in social sciences. And that's reflected in the way I chose to focus this discussion, particularly highlighting gender and socioeconomic differences in modifiable risk factors for dementia. And I will try to make the case that it can be misleading to separate the two, and it's very important to look at the two together. So the material I'll be discussing in the next 15, maybe 20 minutes really draws very heavily on publication together with Elina Suzuki, whose contribution I want to acknowledge. And this is a part of an edited book published by the Women's Brain Project. And I would recommend the book to anyone who has an interest in the topic of sex and gender differences in Alzheimer's disease more general. It covers a series of topics from primary research to clinical research and practice, and to a social science and policy-oriented perspective to dementia. So I have no conflict of interest to declare, but maybe in the interest of honesty, I should say that my background is in economics. I am a health economist by training, and I have no doubt that will become apparent as I talk. So best to be open about the perspective that I'm bringing to things. So let me start by going back to a statistic Professor Lynch mentioned, that 2 in 3 cases of people living with dementia today are actually women. And in Europe, at least, the estimate is, or the most recent I'm aware of, that around 8.8 million people live with dementia, and close to 6 million of those are women. So indeed, 2 in 3 likelihood for people living with dementia to be women. Now, the most common explanation I heard for that as a social scientist has generally been, well, that's probably explained by the survival advantage of women. And it is true that if we look in the general population, the gender ratio is fairly balanced, maybe somewhere around 51%, 52% women and 48, 49 males. But as the population progressively ages, if we look at higher age groups, particularly the very old, so 85 plus, we see that the gender balance is very strongly skewed, close to the same ratio, 2 women for every 1 man. And yes, this is probably part of an explanation, although likely not an explanation in itself, because age is not a cause of disease. But it's certainly, as I think Professor Lynch also highlighted, an unsatisfactory and insufficient explanation. And to explain what I mean by that, I'm going to rely on a fairly stereotypical metaphor of the iceberg. So when we look at the prevalence of dementia, what we're looking at is health outcomes, functioning, general well-being. And if we're trying to address this, if we're trying to reduce this prevalence, if we're trying to reduce the burden of dementia, what we really should be doing is look at the dynamics and the processes that these outcomes are grounded in. So I would highlight here access to care. I would highlight here health status. What are the underlying conditions? What are the pathologies, the biological factors? And even more profound than this, what are the risk exposures that increase the likelihood of a specific disease or a specific condition developing? And this is really the perspective that I'm going to try to take in this talk. That if what we're interested in is reducing the burden of dementia, then the best tools we have at our disposal are prevention-oriented tools. Now, I hope there will be a disease-modifying drug very soon, but until that happens, it actually happens to also be the only tool we have at our disposal. So let me focus on that, on reducing risk factors for dementia, and importantly, modifiable risk factors, because there is a growing and I think very convincing body of evidence that shows there is a very substantial contribution of such modifiable risk factors, so factors that allow for intervention and for modification throughout the life course. And it is very impressive to me that if we correctly address up to 12 modifiable risk factors, we could, in principle, reduce the prevalence of dementia by 40%. And that is an enormous amount, and it needs to sink in. But really, there is enormous scope for intervention, and I think it's a very strong case for investing in a global dementia prevention strategy. So as far as scope for prevention goes, I think this is more than significant. Now, my focus in this talk is also on inequalities. So not only thinking of the process, but on top of it, layering the concept of differences between groups. So if we see higher prevalence of dementia among women, we should ask ourselves, where in the process, where along the iceberg these differences between groups start to seep in, if not everywhere. And now the 12 modifiable risk factors, and in most modifiable risk factors that are known, there is increasing evidence, I would call it very convincing evidence, that there are clear gender patterns. What is even more, there's a parallel body of evidence that shows socioeconomic status measured alternatively as income, wealth, or educational attainment also displays extremely pronounced inequalities. And this is not indifferent because there is a very significant overlap between low socioeconomic status and female gender in most European societies. So at the intersection of the two, of gender and socioeconomic inequalities, we find growing numbers of women who are exposed at very high risk of dementia and have very limited support to mitigate it. So throughout, I'm going to try to follow these two threads of gender and income with the mention that the likelihood of women being overrepresented in the lower income and lower socioeconomic status groups is very high. And now the picture on the left is taken from the Lancet Commission's report, which I think is familiar to everyone. It's a wonderful picture, but it has the disadvantage of not fitting well PowerPoint slide. So on the right-hand side of the screen, I've tried to display it more clearly. And because of the limited time today, I'll try to present some overview of the evidence on only 3 of the modifiable risk factors throughout the life course: education, hypertension, and depression, which was again also mentioned by Professor Lynch. Jumping right in, this is some data again from Europe on self-reported high blood pressure. And by self-reported, what is meant here is that respondents were asked to say if they have been diagnosed by a qualified physician with high blood pressure or hypertension. And all I need to explain in these 2 graphs is that on the left-hand side, the orange circles represent women and the green ones represent males. And on the right-hand side, the lighter colored circles represent lower education groups and the darker colored circles represent higher education groups. It's very obvious, it doesn't take a statistician to see that in virtually all countries we have here, represented here, women are more exposed to the risk of hypertension. More women have hypertension than men. And also, but there are a few differences. So Germany, Ireland is one of them where there seems to be a very small gender difference. Again, this could be partly explained by difference in access to diagnosis between genders. What is absolutely striking to me is that there is no question where the disadvantage lies when we look at educational achievement. So everywhere we look, there is a very significant difference in prevalence of this risk factor between high and low education groups. And I will draw your attention to the fact that the scale of the 2 graphs are different. So the length of the bar on the right-hand side graph represents a larger difference than the length of the bar on the left-hand side one. Now, I've showed you data for low education. The same, the graph would look very, very similar if we had chosen to display it by socioeconomic status, so say income or wealth. But what is very interesting is that for hypertension, there is actually evidence, fairly good evidence, from strong study designs that show an intersectional impact of gender and socioeconomic condition, which means that the strength of association between socioeconomic status and likelihood of getting the hypertension, being diagnosed with hypertension, is higher among women. Women are more vulnerable to the detrimental effects of poverty and of low socioeconomic status than men are. On top of that, consider an additional layer that inequalities in outcome are determined also by inequitable access to care. And there is, again, very convincing evidence that's summarized in the chapter that women and individuals who have higher education attainment are more likely to manage and control hypertension once diagnosed. But at the same time, women and lower income individuals are less likely to be diagnosed, especially in a timely manner when treatment would benefit them most. So consider how these inequalities not intersect, but overlap and enhance each other's effect. A quick overview for depression as a risk factor for dementia, with the caveat that depressive symptoms are recognized as prodromal symptoms because they co-occur so frequently in the presentation of dementia. Nonetheless, I think there is by now consensus that they also act as an independent risk factor, but it's a complex set of interactions, again, because depression can have very strong associations with social isolation, which is an independent risk factor for dementia itself. Similarly, with the lack of physical activity, also a very important independent risk factor. All 3 of them particularly relevant in late life, so for older individuals. Now, dementia prevalence in Europe, extremely marked and very, very clear gender gaps. And here I've displayed the data by age groups, really to make the point that virtually everywhere above the age of 45, so this is middle adulthood, women have a much higher prevalence of depression than men. And the difference is extremely pronounced in later life. And you can see comparatively in this graph how much higher the prevalence of dementia is among women, even women age 45 to 54, as compared to men virtually everywhere in the in their life course. Again, this is prevalence of self-reported diagnosis by a qualified physician. So we should consider here whatever biases may be introduced by lower diagnosis rates among men or lower willingness to report a diagnosis. Nonetheless, there is a clear gender dimension to dementia throughout the life course, and especially so pronounced in older age. Now, the same can be said about socioeconomic status, and if at all, the effect here is even more pronounced. So depression is everywhere more commonly reported by people with lower socioeconomic status, and it makes no difference whether we measure it by education or attainment or income. A very striking statistic to my mind is that based on EU data, the lowest income group, so the poorest quintile in an income distribution, are more than 2 times as likely to report chronic depression than those in the top income distribution. Now, this is a really very high effect and very worrisome. And again, very much as was the case with hypertension, there is increasingly stronger evidence showing that income inequalities are more pronounced among women. Income inequalities, I should say, in depression or in reporting chronic depression. So once again, this suggests very strongly that women are more vulnerable to the detrimental mental health effects of poverty, of deprivation, of everything that comes with the pressures of being part of a low socioeconomic group. Making the same point just with data, as before, women are the orange circles, men the blue. Poorest individuals are displayed by lighter colored circles, and richer individual by more intense colored circles. And here really the point is that you can see by looking at most countries in this graph that it is really poverty rather than female gender that drives the very, very strong inequalities. So you can see that for most countries, the light blue circle actually lies to the right of the dark orange circle. So poorer men are in a worse situation than richer women as groups. Yeah? So these are averages, of course, and this comes with its own biases. Exceptions are Portugal and Bulgaria. But by and large, the strongest effect we see, especially with respect to depression, is an effect that's linked to poverty, that's linked to low socioeconomic status, on which gender overlies complicating and worsening manners. Now, to end on a positive note, or going towards a positive note, education achievement, the one risk factor in early life that has been very consistently shown to be linked with dementia risk. What are the mechanisms here? Well, I'm not best positioned to say, but there is a growing consensus that a lot of these differences might be related to cognitive reserve, which help protect against some of the biological changes in the brain that come with Alzheimer's pathology and dementia more generally. Now, there is a meta-analysis of available evidence that suggests each additional year of education can reduce by 8% the risk of developing Alzheimer's disease. Impressive in itself. And where are we on education achievement? Well, this is the one field where there have been enormous gains in terms of reducing the gender gap to the point where it is disappearing, if not reversing. So in many high-income countries, the gender gap has reversed. There are more female graduates of high school and higher level of education than men. This varies by field, but by and large, it's a consistent result. And it's harder to display with data, but we can look at different age cohorts, and this gives us a sense of what has been happening. So you can compare the cohort that was 25 to 34 years old in 2018 versus the cohort that was now entering old age in 2018, that is 55 to 64. And you can see again this reversal, whereas the rate of upper secondary education attainment or higher was higher for men in the older cohorts. This has reversed in OECD countries, and now it's more women who manage to attain this level of education. Similarly, for the G20 countries, so this includes also countries that are considered middle income or upper middle income countries, showing again there that the gap has reversed. It's much smaller, but there has been a progressive catch up, and in many countries, more women have higher education attainment, or higher than a secondary education attainment than men. Now, this is a very good story, clearly a success story, but it has its dark side in that, once again, if we do keep in mind that separating gender from socioeconomic status is dangerous, then, you know, it's important to also keep in mind that while we've made considerable progress on gender inequalities in education, progress in reducing socioeconomic gaps in education attainment are very much similar to what they used to be. The graph here displays the likelihood of educational attainment depending on the parent's education, very much showing that if at least one of the parents has attained tertiary education, the likelihood of an offspring to attain tertiary education is somewhere around 63%. But if neither parent has attained upper secondary education, then the offspring has around a 13% chance of getting to tertiary education attainment. So again, a very, very significant gap that we have not managed to make much, if any, progress on. Now, let me try to bring everything together because it was a lot and might be confusing. This is really all just to give you a glimpse in that there are pervasive and profound gender inequalities in modifiable risk factor for Alzheimer's. I've showed you only a few, but this is the case for virtually all factors. There are very few examples where the gender gap is inverse, and one that comes to mind is alcohol intake. But by and large, women are disadvantaged throughout the life course and are exposed to much higher risk, which is very likely to contribute to explaining why the prevalence of dementia is higher among women. Very important point I hope to take is that gender and social class inequalities intersect. This makes women not only overrepresented in lower income groups, but more vulnerable in their health outcomes and health status to the detrimental impacts of low socioeconomic status. The optimistic story is that gender differences in some risk factors, we've talked about education, are being attenuated, and we will see an effect as more recent cohorts move towards old age. This shows that it's possible. This shows that progress can be made if sufficient investment is committed to addressing these problems. And I hope I have convinced you that a successful prevention strategy must take a population health approach and must really build on reducing inequalities in risk factors. So if we manage to address these inequalities, if we manage to address the risk factors for the 2/3 of people living with dementia who are women today and are likely to be women in the future, if we fail, this is an extremely large gain. And this, of course, I haven't had a chance to go into details, but absolutely needs to be paralleled to understanding and reducing inequalities in access to treatment and care. This includes diagnosis. I'll stop there because I think I've already overshot my time, but please write with questions and please consult the book. It's much more detailed than I could be here. Thank you very much.
That's great. Thank you very much, Stefania, for a very clear presentation. And actually, there's a question that came in there, I'll call it out too, just in relation to hearing loss, because I know you had that slide on all the different modifiable risk factors. So the question is, in which sense hearing loss constitutes a risk factor? Can it be modified introducing a hearing aid when hearing loss starts? So I don't know if you had any comment on that.
Yeah, Professor Lynch might be better positioned to answer this than me, but I can tell you from what I know of the literature that it actually is one of the risk factors that has the highest effect. So I think somewhere close to 5 or 6% reduction in general prevalence. It is my understanding that it has to do primarily with how stimulated, how much cognitive stimulation a person gets, or the reduction in cognitive stimulation as hearing declines. And exactly, it can be addressed by prevention. Yeah, we can understand what are the risk factors to hearing loss and address them. It can be addressed by diagnosis, and it can be addressed by very inexpensive hearing aids. So this is already highly accessible, very inexpensive assistive technology, which is unfortunately not included in public health packages in most European countries. Nonetheless, it is extremely accessible, and I think, or at least I'm aware of some studies that are showing high cost effectiveness, particularly because of this, because it's low cost, but potentially very high impact. Professor Lynch might want to add to that. As I say, I don't feel very well placed.
Well, one of the things that I'd just like to say with regard to this is that one of the things that has been found to be extremely good in terms of slowing the progression of the disease and reducing risk is learning the piano. And the one of the things about the piano, as everybody knows, is that when you're playing, you have so many things to consider. So first of all, you have motor function because your hands are moving, and sometimes your feet if you're using the pedals. Secondly, you're looking at something that's incomprehensible unless you know a little bit about it. That is black and white dots on a page in front of you that you've got to see, translate into a motor function, because you have to translate these dots into notes and actually be able to produce the notes on the keyboard. But in addition, and this is probably the most important point in regard to hearing, is that all the time you are listening, correcting, and you have that feedback going on at the level of the thalamus in the brain, which coordinates all the senses. So all these senses that are being stimulated in the course of this business of playing the piano, or indeed the tin whistle, if you felt like playing the tin whistle, it's just incredibly important. And losing the impact of one of these senses is really what we're talking about here. And all of them can be, to some extent, at least, corrected by assistive technologies, as Stefania has just said. So it's a really nice story to know that you can do something to push the disease down the road. Just learn an instrument.
Absolutely. And actually, we ran a brain health webinar series last year, and all the recordings are on our YouTube channel. So if people are interested in that, I know that came up actually on one of the talks about amateur musicians. There was some study comparing professional musicians with amateur musicians, and amateur musicians had that advantage because their brains had to work so much harder. Well, that's the theory, that there was more of an advantage cognitively. So if people want to have a look at those webinars, they're available on our.
But there's a lovely story. I mean, musicianship is one thing, but it's entirely different story when you're not musical in any way at all, but you still see the benefit of this. And so there's a really nice study from a few years ago where a bunch of older people were split into two. One that learned the piano and practiced for 45 minutes each day for, I think it was only 8 weeks. And now it could have been longer, but anyway, for a very short space of time. And then all the other group did all sorts of things that are normally considered to be very stimulating, like meditation, like yoga, like golf, and etc., etc. And the people who learned the piano came out far better in terms of cognitive function. So, I mean, you think I was pushing the piano. I don't mean to do that, but what I'm saying is that there's a really good message out there, that you can do something. You know, it's lovely.
Yeah, absolutely. Because I think when we hear about these modifiable risk factors, and people are thinking about risks, and to have the balance of, well, what can we actually do about it? How can we try and prevent dementia? It's, as you say, an important kind of public health message, if you like. And actually, just to mention as well, there is a European study called SenseCog, and Professor Ilinica Leroy, who's in our research network, and she's based in Trinity College and St. James's Hospital. She headed up that European program, and that was looking at intervention around hearing and vision. to improve cognition. And they're now actually, there's a project called Saints Cog Care being rolled out in Ireland, which is looking at that intervention for nursing homes in Ireland. So very exciting. It's just started that project. So a very exciting time looking at hearing and vision. And as Stefania and Marina both said, you know, something as simple as a hearing aid, you know, can make obviously a big difference. So that's great. So sorry.
Can I add a thought to that, Carol? Also, because I see another question in the chat that is about what individual risk a specific person has with that you know, can recognize a series of risk factors. I really want to underline that there is an important difference between kind of an individual risk reduction strategy and the population-wide prevention approach. And a lot of what I was talking about really takes this population perspective. But I think the point Professor Lynch was making before about what each individual can do to reduce their individual risk is also a very important part of the story. So yeah, it's not so immediate to translate most of the data I presented or the messages that are relevant at policy level to the individual level. But I do think there are more and more available materials about what each and every one of us can do at any age to reduce our risk. And I would never, and I don't think anyone would venture an actual percent to quantify such risk. But it's certainly, there are many specific strategies that help us reduce whatever risk we start with on a genetic basis, on a socio-economic basis. And I think that's an important focus to support each and every one of us to improve our own outlook.
Great, thanks. And yeah, I don't know if you're kind of touching out there, but in terms of the genetic element to Alzheimer's disease, you know, I don't know if either of you wanted to comment on that. There's a question about what percentage would I have of getting dementia when my mom and her sister has it. I also suffer from depression and a heart disease. So obviously this person is looking at their own risk factors, but certainly the genetic side of things is you know, usually just a small element, isn't it? I don't know if either of you want to put a percentage or figure on that, but my understanding it's a small contribution usually, unless it's a particular type of Alzheimer's.
Well, we know that the vast majority of sufferers, that is possibly close to 95%, suffer from Alzheimer's that is a result probably more of age than genetics. There's a very small proportion of cases where you have what's called early onset dementia, which is a genetic disorder. So of course, any one person in a family where early onset is a problem might or might not be, might or might not have the same genetic makeup as their parent or sibling or whatever. So from the point of view of that question, it's really not possible to give an answer in terms of percentage. That would be something that would have to be figured out after genetic testing, for example. But I noticed that person says they suffer from depression and of heart disease. Well, there are certainly risk factors, but when they're controlled, you have a different scenario entirely. So the thing is that the trouble is when they're not controlled. So if you keep your heart disease under control and the depression under control, then you almost eliminate the risk that's associated with them. So that's a nice story. And the other thing that's well worth pointing out is that exercise is hugely beneficial. So that person might consider an exercise program. We know, for example, in our mouse model. Now, this is a mouse model, I have to hasten to add, but we know that in the mouse model, if we get the mice to exercise, we can actually get those microglia that I was talking about to become less inflammatory. And we also seem to be able to kickstart them in terms of their metabolism. So exercise has huge benefits directly on the brain and on these cells that might cause inflammation. So I would advocate you know, an exercise program for most people, as much as you can do. Clearly, it's going to be helpful. That's what I would say about that. It's not all bad news. There are lots of things, and Stefania pointed out that modifiable risk factors can reduce your risk by 40%. It's huge, huge.
Yeah. And again, if people are interested in learning more about exercise or nutrition, we have those brain health webinars from our series last year. So I think it complements a lot of what we're talking about today. There's also a question about whether Alzheimer's disease is more common in people with autoimmune disorders, example MS. I don't know if you're aware of any research around that.
I mean, certainly we know that MS is more common in women than in men, for sure, whereas Parkinson's, for instance, is more common in men than women. But I can't recall any evidence that suggests that autoimmune disorders is associated with a higher prevalence of AD. Sorry, I don't know the answer to that.
No problem. Okay. And then there's a question about, it's an interesting question. Everything I read or watch these days seem to indicate that women are ignored to a greater extent by the medical profession, which has historically been geared towards men. How much lobbying or influence do dementia specialists have in seeking to change this imbalance? I don't know if you have a comment on that.
I think that
I'd say that it would take an entirely new webinar, or maybe 2 or 3, to address this. I would encourage you to follow the work of the Women's Brain Project. So this is an association that is trying to do exactly this, to raise awareness of sex and gender differences and a lot of structurally embedded sexism in everywhere, from clinical research to clinical practice, diagnosis, everywhere where there are gender differences, and they do signpost evidence that is coming on a regular basis. From what I know of dementia, women get diagnosed later in the disease progression, partly because, it could be partly because the symptoms that are used to identify dementia have been developed primarily on clinical studies that included men. There's still a very, very unbalanced gender distribution in many clinical studies that are being carried out for a range of neurocognitive studies. Same for treatment. So there are issues with when women get diagnosed, to what extent they get the same quality of treatment, to what extent can they afford the same quality of treatment. But there might also be biological differences that are poorly understood because they are not properly studied, because the makeup of the samples that have been predominant in clinical studies is gender biased. And Professor Lynch might be able to tell you more, but I have also heard absolutely mind-numbing evidence that this gender imbalance that favor males is replicated in animal model studies. What exactly the reason there could be, I truly, it's beyond me, but yeah, it seems to be a very systematic bias that seeps through each and every hole.
That's true. The bias has been there in the past. In animal studies as well, and by and large it was because including female, for instance, mice, it complicates the issue because you've got the cycle, cycle and hormone changes that actually could contribute to differences. However, the good news is that every funding agency now insists that males and females are used in equal number, and that researchers will not get funded anymore unless they have included males and females. So I think that if you look at the literature over the past 5 years, you can see that there's far more emphasis on ensuring that you've got equal numbers of males and females. And this, I think, is only going to, I think it's going to just continue this news.
Great, thank you for that. And so I've just put up a slide there for people just in relation to some websites and resources that people might want to look up. And I'm also just going to launch a poll now, just to get some feedback from you, if I can find it on my screen here.